Identifying Neuroanatomical Phenotypes in Mental Illness by Adelina Boda. The purpose of this study is to identify convergent and divergent cortical phenotypes across autism, ADHD, and schizophrenia. The experimental hypothesis aims at discovering if autism spectrum disorders, ASD, attention deficit hyperactivity disorder, ADHD, and schizophrenia share neuroanatomical variations. Researchers used measures of cortical anatomy to estimate spatial overlap of neuroanatomical variation using univariate methods and developed a novel methodology to determine whether cortical deficits specifically target or are enriched within functional resting state networks. Shared genetic risk factors across autism spectrum disorders, ASD, attention deficit hyperactivity disorder, ADHD, and schizophrenia include perinatal hypoxia, antenatal maternal and postnatal infections, and maternal smoking during pregnancy. Shared phenotypic traits show neurobiological indicators of brain connectivity abnormalities across these disorders, indicating altered default ventral attention, sensory and executive networks in individuals with ASD. The default mode, frontoparietal and ventral attention networks in those with ADHD, and deficits in all of the mentioned networks in those with schizophrenia. Shared mechanisms between these disorders is indicated by common neurocognitive and clinical deficits in individuals with ASD, ADHD, and schizophrenia who share similar deficits in social cognition, language abilities, and cognitive function. Individuals with ASD and schizophrenia share somatomotor deficits, while those with ASD and ADHD share impairments in executive function and emotion regulation. Those who have negative symptoms of schizophrenia show similar patterns of social withdrawal and poor eye contact. Additionally, up to 75% of individuals with ASD have shown ADHD symptoms, including attention and motor control problems and hyperactivity. Given the overlap in risk factors and clinical features, researchers sought to determine if there is a convergence of structural brain alterations across these disorders, postulating that these alterations may take two different neural topologies. The first form would show a convergence of neuroanatomical deficits localized to specific neuroanatomical subregions, indicating focal neuroanatomical differences that colocalize within patient groups compared with a control group, thereby resulting in group differences that can be identified using a parametric statistical test. The second representation is a distributed topology, or a set of neuroanatomical group differences that may colocalize within a more broadly defined area, such as resting state networks, RSNs, thereby possibly impacting brain function related to network function, where subtle alterations may exist at a local level, yet these alterations may be systematically pervasive across specific neural pathways, resulting in preferential alterations in specific functional networks of the brain. Studies have noted shared structural abnormalities with the anterior cingulate cortex and insula being identified as locations of structural and functional changes corresponding to the salience or ventral attention network. Thus, researchers expected to find shared neuroanatomical differences in the anterior cingulate cortex and insula. To determine the validity of their findings, researchers investigated whether the neuroanatomical anomalies they found aligned with previous findings from functional neuroimaging studies, and therefore performed this analysis using the NeuroSynth database, aiming to build structure-function homology between the researchers' structural findings and previous functional MRI or fMRI results. Researchers attempted to examine cortical topography of cortical thickness, CT, and surface area, SA, across patients with ASD, ADHD, and schizophrenia by performing 3 different analyses to determine if deficits across disorders could be localized to a subset of neuroanatomical structures, thereby examining neuroanatomical differences between cases and controls across all 3 disorders and exploring neuroanatomical convergence using conjunction analysis examining shared deficits. Researchers examined whether the neuroanatomical results derived through the above analyses aligned with functional findings using the Neurosynth database http://neurosynth.org/ as it enabled large-scale automated meta-analysis of previously published fMRI studies by searching the Neurosynth database in March 2016 using the terms autism, schizophrenia, and ADHD, resulting in 148 studies of ASD, 73 studies of ADHD, and 249 studies of schizophrenia. Given that the disorders of interest are thought to be the product of compromised neurodevelopmental processes, researchers developed a technique to examine network-level enrichment of neuroanatomical deficits, assuming deficits would be pervasive within a functional network but would not necessarily follow the mass action type of deficit, and therefore adapted statistical methods from GSEA tests to determine whether sets of individual genes are collectively enriched in biological pathways. Across disorders, patients with ASD and those with ADHD presented with significant enrichment in the default mode network, but those with ASD showed positive and those with ADHD showed negative enrichment. Although the direction of local change differed, both showed convergence at the network level. Patients with ASD and those with schizophrenia both showed significant enrichment in the frontoparietal and limbic networks, but those with schizophrenia showed negative and those with ASD showed positive enrichment. Global correlations for cortical thickness and surface area indicated that patients with ADHD and those with schizophrenia showed the greatest similarity in terms of brain regions affected. Patients with ASD had negative correlations for cortical thickness and surface area with both schizophrenia and ADHD, indicating that ASD affects different brain regions than ADHD and schizophrenia and in opposite directions, thickening in patients with ASD versus thinning in those with ADHD and schizophrenia. In examining surface area enrichment, researchers found greater convergence across disorders than in cortical thickness and in patients with ASD. Significant positive enrichment was seen for all networks except the default mode network, suggesting network-specific patterns of cortical overgrowth. Patients with ADHD presented network-specific directions of enrichment with significant negative enrichment in the somatomotor network and positive enrichment in the visual network. The visual network covers almost the entire occipital lobe and divides into smaller networks in the 17-network cluster definition, indicating that although all 3 disorders do not affect a specific region within the visual network, different subcomponents of the extended visual network are affected in patients with schizophrenia compared with those with ASD and ADHD. Altered neuroanatomy in psychiatric disorders has traditionally focused on finding reductions of gray matter in distinct regions of the brain. However, researchers showed that by studying multiple psychiatric disorders simultaneously, patterns of altered brain structure extend beyond simple reductions and found that deficits across disorders may arise through coordinated, widespread alterations within functional resting state networks responsible for dynamic cortical processing of complex human traits. Researchers found evidence of divergent local measures of gray matter and poor correlations between disorder-specific effects toward convergent network-wide alterations across ASD, ADHD, and schizophrenia. The heterogeneity of their findings call for revised systemic approaches in studying these disorders and conclude that structural neuroimaging in ASD, ADHD, and schizophrenia should move toward investigation of multivariate patterns and establish multimodal homology using novel statistical methods because global disruptions in brain structure, rather than strictly localized changes, must be taken into account for greater insight into the association between altered structure and neurocognitive deficits in psychiatric disorders. Cortical anomalies were preferentially enriched across functional networks rather than clustering spatially with cortical thickness, showing significant enrichment between patients with ASD and those with ADHD in the default mode network, between patients with ASD and those with schizophrenia in the frontoparietal and limbic networks, and between patients with ADHD and those with schizophrenia in the ventral attention network. These networks enriched in cortical thickness anomalies were represented in functional MRI results. Reference: Park M.T. M., Rosnahan A., Shaw P., Gogte N., Lurch J.P., and Chakravarti M.M. 2018. Neuroanatomical Phenotypes in Mental Illness, Identifying Convergent and Divergent Cortical Phenotypes Across Autism, ADHD, and Schizophrenia, Journal of Psychiatry and Neuroscience, JPN 43-3201 to 212, page numbers.